of the last. So let's finish this up and then a couple other things. All right, so just as a review, we went through phase 1, 2, and 3 clinical trials. So after a drug comes out of animal trials and it looks like it's safe, it goes into, somewhere between 20 and 100 humans who are healthy, don't have anything wrong with them, and we get ADME in humans, and we poke them and prod them and ask them lots of questions, and we're looking for safety in humans. Then it goes, assuming it gets through that, it goes into phase 2 clinical trials, where now you have a few hundred people who have the disease, but they don't have anything else wrong with them. So now you're looking for efficacy. Still always looking for toxicity. And then phase 3, now it's going to thousands of people, placebo-controlled studies, and they're going to be long. They might go on for quite some time. They might take a couple of 3 or 4 years of phase 3 clinical trials, depending on the drug. It really depends on what the FDA needs. Okay, and then phase 4 is once the drug is approved, it goes out into the market, but they have to watch that. And there have been cases where stuff came up that in phase 4 clinical trials, because again, if we test, let's say we tested 20,000 people even in say 4 or 5 phase 3 trials, tested 20,000 people, but it's a drug that's going to go out and everyone's going to take it, like something like Lipitor or an antihypertensive for high blood pressure, and it's going to go to tens of thousands or hundreds of thousands or even millions of people are going to take this drug. There's going to be things that slip through the cracks that just were never caught in a test. That's why it's very important that phase 4 trials take place as well, that they continue to monitor. All right, a couple other considerations. The FDA doesn't just approve the drug works, they also have to approve the manufacturing process. So how is the drug made? This is called good manufacturing practice. So the GLP is good laboratory practice, that is preclinical. GCP is good clinical practice, that governs clinical trials. Good manufacturing practice manufactures how the drug is made. Most people don't realize this, but the very first time somebody made the molecule that's in, we'll pick a drug like Lipitor, the first time somebody made that particular molecule, they probably made milligrams of it. Very small amount in a test tube, very, very little bit. When Lipitor is manufactured, it is not made in test tubes, it's made in vats the size of our classroom. So they're very, very big, they're making tons at a time. So why don't we do the same chemical reactions? Well, let's say that we did the thing in a test tube, and to make it, we had to do a reaction that gives off a little bit of heat. But a little bit of heat, big deal. But if you bring that up a billion-fold, now it's a lot of heat, it'll burn down the building. Or what if, well, we had to work with this toxic chemical, but we do it in a fume hood, no big deal, it's easy to remove. Well, now you're going to billion-fold it, you're going to go up from milligrams to kilograms, right? Or milligrams to tons. And you just can't work with molecules like that. So typically they have to find ways to make the drug that you can make it the same way every time, that it can be made safely, that it doesn't cost ridiculous amounts of money. I'll just give you an example. A guy I know I used to work with when I taught this class, he was the lead chemist on Lyrica, which is a drug for nerve pain. It's used a lot in diabetic neuropathy. And his group developed that drug. They didn't discover it, but they figured out the way to manufacture it. And when it was first made, they did a lot of stuff in organic solvents, and you guys know what organic solvents are. And those can't be thrown down the drain, right? Those are nasty chemicals. He figured out a way to do it in water, and then there was almost no waste. So he found a very nice environmental way. Environmental considerations are big, safety considerations. But the most important thing is that you have to demonstrate that you can make the same drug every time. So when you go to the store and you buy Tylenol, you're assuming that every pill in that package is exactly the same, right? That not one of them doesn't have more than the other, that they're all the same, it's all the same molecule, all the same extra ingredients, all the extra ingredients have to be approved, the packaging has to be approved, all sorts of stuff. Right? It's all stuff that has to be considered, and that process happens kind of in parallel with the other testing. Generics. So generics come in after the patent. So, and the next slide talks about patents. But the company that develops the drug has exclusivity. Generics come in afterwards, and of course the price goes way down. Generics still have to be FDA approved, but since the drug was already FDA approved, the generic company doesn't have to go through all of that testing because it's been done. pretty much for generics, they just have to demonstrate that they're making the same drug and they're making it that they can make it the same way. So there's a lot of the other stuff's already done. They just have to demonstrate that they are indeed making, so that the Tylenol they're making, the acetaminophen, is the same acetaminophen. All right, vaccines actually fall under what are called biologics. So the FDA actually approves 3 things: drugs, biologics, and devices. So a device would be like a heart monitor, crutches, things like that. Biologics are things like vaccines. They have a little bit different process. They have to show that it works. They still have to do that. They have to definitely, especially if it's an injectable, has to demonstrate that it's sterile. They have to be careful about allergic responses. And we can talk a little bit about vaccines, and we talked about it before. Vaccines are typically heat-killed or disabled bacteria or virus. So it actually has, so to speak, the disease in it, but it's an inactive form. But they have to demonstrate that it's inactive and that they can do that every time. They have to demonstrate that let's say each shot has 10 million viruses, dead viruses in it, that each shot does indeed have 10 million dead viruses in it. Right, that kind of thing. A big thing with biologics, because they're biological entities, is allergic responses. So they have to look out for stuff like that. It's a little bit different process. You don't have to discover the drug from nothing. And a lot of times vaccines, there's a lot of stuff that's already done. They can just put a different thing in kind of the same package. So it can be a little, like when they're talking about the COVID vaccine, they said, well, it'll probably take about a year. That's not 15 years. And that's because you're not starting from scratch. Some other points to talk about: off-label use. So if you look at the slide very early on, right, that has a lot of words on it where it talks about a lot of drugs that are being worked on for COVID-19. A lot of those drugs are off-label use, meaning they're actually drugs for other things, but they might be used for this. Okay. Well, if it's a drug that's already been approved, it's already got a safety profile that's been approved, so they don't really have to do quite as much safety work. If they're going to do different doses or different formulations, so they're going to mix it with different things, they have to show that that's safe. But they don't have to show the drug is safe because that's already been done. But they do have to show efficacy. Viagra is a great example. Viagra was originally created to be a drug for blood pressure, and it had a very interesting side effect. So that's an example of an off-label use. And then the money part, big pharma is held up as the evil empire because they make a huge amount of money, and they do. They spend a lot of money to develop drugs, but they are making a profit. They're making a lot of money, and that's a different discussion for a different class. I am not going to be someone who is going to make excuses for that industry, because I think that just like, they do a lot of stuff, but people do live a lot longer because of that, so there is that as well, right? People used to die in their 30s, 40s, 50s. And people didn't even live to be my age back in the 1930s. So there's a lot of that. But the profit part of that is something that really does need to be addressed, because it's kind of getting crazy to a certain degree. To give you an idea of how patents work, patents give a company 20 years of exclusivity from when the patent is filed, or I think when the patent is approved. Typically, they do the patent before the drug is out, because other people are working on the same technology. So that's a trick. What you get for a patent, though, you get exclusivity for 20 years, but you also have to tell everybody how you do it. That's why generics can come in. So they'll say, here's the drug, here's the data, it's public information, and once the patent is out, it has expired, there's nothing to prevent other people from doing it because they actually told you how. Anyway, that should give you a feel for the drug discovery and development process. Yeah, obviously, it's very short because we have a lecture or two. So at this point, that's it. You're just going to there's a homework over this. For the class, there's exam 4, and there's the final. I'm really proud of all of you for adapting to this format. It was difficult for me. I'm sure it was equally difficult for you. Those of you who are still here, you did a great job. And obviously, I'm going to teach this class again, and it's going to be a little bit different because we're just making it up as we go along. But way to go on being adaptable and working with things, and I know it was difficult for you. But I really hope you really learned a lot in this class in spite of how a lot of the things played out. Certainly with what's happened in the world, while we're with this pandemic, makes this class a lot more relevant than it sometimes is. Anyway, it's been great getting to know you as well as I did, and when we're all back on campus someday, feel free to come by, and I'd love to say hi to you. Catch you later. Best of luck to you.